mic button is on the bottom right of your screen. There's a little microphone button. So if you press on there, we should be able to hear you. So the unmute button is all the way on your right. There's a little microphone symbol on the right corner on the bottom. So if you press on there, you should be able to speak.
Hello, everyone.
Hi, Victoria. How are you?
I'm well, thank you. Katarina, Jamie, Lizebona. Is that how I pronounce your name?
The unmute, you still muted because your mute symbol is still up. So if you could try to press the button again, then it should be working. Yeah, it's still your mute symbol is still up. We have, I don't know, did you ever have the issue, Victoria, with this one that says she unmuted, but her mute button, like the symbol is still up and we don't.
Yeah, I suggest that you try leaving and coming back, and that should fix it.
Maybe the app is somehow stuck.
Ever since they did the spatial audio, then people occasionally still have that problem. So I would try that. I would try leaving and then coming back.
Yeah, you're still muted. The mute button is still on.
Okay, good.
Good. Let me add the topics.
Oh, good. We've already got the article pinned. Hello, welcome back. Can you unmic or unmute? So when you click the mic icon on the lower right-hand side of your screen, nothing's happening, is that it?
No.
Nothing happens. Okay. My next question is your app recently updated? I see that you're new on the app, so I would think it would be the most recent version.
Should be, right? She just joined, so. Are you on your home Wi-Fi network or at your university? Because
that's a great question. Yeah, if she gets off Wi-Fi. When you shop at Fred Meyer, you can double your. Oops, we don't need to hear that. Welcome back. Yes.
Did it work? Oh, great. Hi. What was it? Was it the Wi-Fi?
Yeah, I'm in uni now.
Yeah, so sometimes the universities block. Okay.
Fantastic. Can you please direct us on how to properly pronounce your name?
Lizebona Augustambot.
Lizebona. Okay, nice to meet you.
Nice to meet you too.
Pleasure to meet you, Doctor.
Hi.
Let's wait a few more minutes to give people time to arrive. Well, thank you, and thank you. Sorry that you had to like come in and get out and figure it out.
That's okay.
Stuff like
Your
this.
paper looks fascinating, Doctor. I'm looking forward to this talk very much. This using sonic waves to do these things is quite exciting. It's kind of very science fiction.
Good to hear that. It's interesting work. It's very interesting.
You know, when you your paper, when you're discussing using sound waves to affect the the molecules and everything, it's quite exciting using something like that.
Yeah, so mechanotransduction has been there for recently.
You kind of went mute a bit.
Victoria, are you
anyone
playing
else?
music in the background? Is someone playing music? I can hear.
Yeah, I am, but I can't hear Lizebona.
Yeah.
Okay.
Okay, let's see now.
Okay, can you hear me now?
Yes, I can.
We can hear you, but your voice is it got very soft. I wonder if you're wearing some it's often best to just speak into the speakerphone instead of using any kind of nice headphone.
Yeah. I was using headphone earlier. Now I removed my headphone.
That's better. Okay, thank you.
I have the same problem. I bought really cool, expensive noise cancelling headphones, and they are not good for Clubhouse for whatever reason. I was very annoyed.
Exactly.
Well, yeah. How is your day? Your winter is starting, right? It's kind of fall.
Yeah, it is cool. It's almost 9 degrees today, but I think around afternoon it will be 21, 22, so it should be okay.
Oh, that's nice. That's pretty good.
Yeah, you see that range 9 to 22 within half a day. So.
Oh wow. Yeah, here the spring has not been very good. It's been pretty cold and pretty bad weather. But I hope, well, soon it will be again hot. That I wish it was spring. It was cooler again. The humidity in New York City gets quite bad. Okay, I think we can slowly start. And then we'll introduce you, and then we can go. I'll introduce you, and then usually
you can give a brief. Yeah, like it would be great if you can give a brief introduction on the audience itself, because I'm not sure how to present it. If should I present like in the sense of a hardcore molecular biology aspect or a generalized aspect? So
but so it would good to start with a very general aspect, and then maybe also more specific.
Yeah, sure. So can I start?
Okay, let me go ahead and introduce you first. So, welcome everyone to the Science Society. We Lizebona August. She will be talking about her really interesting research. And let me give you a little bit of background information. She did her bachelor's degree in biotechnology at the Mar Augustinos College in Ramapuram. and she did her higher education in at the NGM college in Tamil Nadu, and she worked in between as a patent analyst, and then she moved ahead and was a junior research fellow at the Sri Chitra Tirunal Institute for Medical Science and Technology, and later later on she moved to Melbourne, where she is now. Hi, Serena. Welcome.
Hello.
So yeah, thank you so much for coming. And if it's okay, Victoria will ask you first more like a very general question, and
Yep.
and then the stage is yours.
Okay.
Thank you. I'm so glad we got all mics figured out. Hi, Serena. Welcome. Nice to see you.
Hello.
Friends in the audience, Sherry, Kay, Rupesh, Victoria. So, Lizebona, yes, if that's okay, it would be
Yeah.
great to just ask you some, just a general question so that we can familiarize with you as a person as well as on the basis of your research. So, if it, can you hear me okay? Because I have the red bar. Shall I ignore the red bar?
Yeah, I can hear you properly.
Fantastic. Okay,
I
right.
can hear you properly. Yeah.
Okay, that's great. So I'm asking if you can reflect on your experience in childhood or later on and try to see if you can pinpoint a time that you really noticed that you felt a strong connection to science. Maybe, an experience or a class, something that
Yeah.
happens that, yeah, that made you feel like, I'm a science person. This is for me. And then after that, if you can share an overview of the path that has led you to what you're about to present today. So thank you.
Okay. So I'm born and brought up in India. So I used to read Reader's Digest when I was young. And one of the article in I think it's very old Reader's Digest, 1987 Reader's Digest, it had an article uh, about a neurosurgeon. And I started reading about neuroscience. I fell in love with blood brain barrier and brain. So my I think I was studying in seventh when I was reading that. So from that day onwards, I was more interested in knowing about blood-brain barrier, how to cross blood-brain barrier, why brain remains white organ, even if the science have advanced a lot. Um, so that's when I identified. I mean, I wanted to be a scientist or someone who does research in blood-brain barrier from that point onwards. So my whole rest of the academics were something that is always directional towards these kind of studies. Um, After my masters, which I mean, I did my bachelor's and master's in biotechnology, hoping that someday I could do something in blood-brain barrier. After my master's, I worked on nanoparticles, like gene therapy based nanoparticles, and these nanoparticles were targeted towards blood-brain barrier. Had couple of papers out of that research. Then I pursued a bit more hardcore molecular biology studies so that I can gain more experience in antibody creation, monoclonal antibody, so that I can create an antibody that can specifically target blood-brain barrier. So that's my whole interest is in neuroscience and blood-brain barrier, and I joined in Leslie's group because they had a cool project where they were targeting like developing advanced lung and brain therapeutics using acoustic waves. So I was not an engineer, so but I like studying interdisciplinary subjects. So I joined his group, and they were doing amazing job, and they were exploring the biological aspect of this acoustic wave, high frequency acoustic wave. And I started my work in exosomes, which is like a natural nanoparticles. and then stepped into stem cell differentiation. Our primary initial aim was to differentiate stem cells into different lineage. It was not just osteoblast. We were targeting different lineages, but unfortunately we ended up only with osteoblast. But I'm pretty sure with a bit more understanding about this mechanotransduction with acoustic wave, we can create something that can pinpoint towards or go towards blood-brain barrier. Hope that summarize what you asked.
Yeah, thank you very
Yeah,
much.
I
It's really
can't.
fascinating to get a chance to, you know, just learn a little bit more and hear about the process. And I also remember the first time I even heard the phrase
Mhm.
blood-brain barrier. I was enamored. You know, like, there's such a thing. So thank you. And now the stage is yours to offer your presentation, and then generally we'll have a kind of a Q&A after the presentation. So yeah, thank you so much. We really appreciate your time here, and the mic is yours.
Yeah, thank you. Thank you so much for giving me a chance to present my work here. So I'll begin with the basics. Like uh, our human body contains around 37.2 trillion cells, and these cells you can uh, generally classified into 200 different types of cells. And the most amazing factors that all these 200 different types of cells originated from a single cell, an embryo. So scientists were always amazed with this fact: how can a single embryo give rise to different types of cells? And they found out that this is because of a cool group of cells called stem cells, which has a capacity to give rise to different type of cells or differentiate into different type of cells. And there, that capacity you call it as multipotency. So most of the stem cells which we see, like stem cells from bone marrow, stem cells from adipose tissue, all these stem cells have a very innate ability to differentiate into different type of cells. And later we researchers found out that you can differentiate the cells into whatever cells you require and use that as a replacement for any faulty cells in your system. So regenerative therapy based on stem cells can be an answer for every diseases which we can know of, and people have been trying to study these amazing cells and how we can manipulate them so that we can get around all the diseases. And initially, it started with using chemicals or biomolecules so that they can direct the cells into different lineages, say for bone cells or adipose tissue cells or skeletal muscle cells. And even in bone cells, you have different three types. Generally, you can find three types of cells: osteoblast, osteoclast, osteocytes. So, depending on what you add into your cells or what chemical cues or biomolecular cue you give, you can give rise to different types of cells. Um, and later they found out that it's not just the biomolecules or chemical cues that is affecting the lineage of the cells. Mechanical stimulation or mechanical stress can also affect the stem cells differentiation, because in our system, when these stem cells are in our body, it experience lot of mechanical cues as well. For example, a shear stress, or it is confined by surrounding cells. So depending on the number of cells that is surrounded by what sort of pressure or like that push and pull it experienced by the neighboring cells affects the stem cells linearity, stem cells differentiation linearity. So a vast number of these kind of mechanical cues were later studied, and it was found out that whatever mechanical cues the cells are experiencing, it converts that cues into different biochemical signals, which decides its fate. And this whole process is called as mechanotransduction. So basically, cells are really impulsive systems. So it can sense any change in physiochemical environment and adapt and response to these changes. And various forms of mechanical stimulations like stretch, shear, pressure, stiffness, geometrical confinement, topographic cues are sensed by very cool mechanosensors that is present on the cells. And it triggers a change in um, signaling molecules. So the coolest signaling molecules that I can think of is second messengers like calcium, for example, intracellular calcium, CAMP, cyclic adenosine monophosphate. So these are very small molecules, but any change in their concentration in the cell or outside the cells can affect massive changes within the cell profile. For example, its protein profile or mRNA profile. And these changes will affect cellular activities like cell proliferation, differentiation, vesicle trafficking, cellular or tissue organization. So basically, you change the physical environment. You can give you can get an n number of modifications within the cell's activity. So most of the mechanical stimulations as in stretch, shear, pressure, when the cells are exposed to these, whatever outcome we can generate, they have been put in use in various field like gene therapy, where you stimulate the cell membrane become more permeable, and you can allow nanoparticles, and it will allow more nanoparticles to get inside the cells. So these mechanical stimulation have been used in gene therapy. And there are other mechanical stimulations or even physical stimulations like radiation, where you can kill the cells and you can use it as a cancer, as in cancer therapy, and like that many more. But issue is most of this mechanical stimulations, you require the cells to be exposed to these stimulation for a very long time. And say for example in regenerative, like in stem cell differentiation studies, these cells are exposed to mechanical stimulation for around 14 days, throughout 14 days, and it will affect the cell viability. And obviously cells is not in its own environment, so it will affect the final outcome. And this is because we still don't have much understanding about the mechanotransduction involved in each of the stimulation. So we need to understand that, and at the same time we need to develop a system which can where you don't have to expose the cells for a very long period, but you can give a short stimulation, so that it can act as a pre-treatment, and then bring about all these changes which we discussed. So our lab focuses on very cool uh, mechanostimulation called surface reflected bulk wave. So these are mega, similar to acoustic waves, which are of megahertz frequency. So we have a piezoelectric chip which can convert electrical energy into mechanical energy. So we provide electrical AC current onto IDTs that is imprinted on this piezoelectric chip made of lithium niobate. And once you apply this AC current onto this IDTs or interdigited transducers, these get converted into high frequency mechanical stimulation or surface reflected bulk wave. So we can adjust the frequency, say 10 megahertz, 30 megahertz, 50 megahertz, depending on the width of the IDTs and the gap between these IDTs. And once these stimulations are generated, you can transduce these stimulations onto different particles. Say, for example, if you have a liquid or materials that is dissolved in liquid, if you apply these mechanical stimulation into these liquid, you can nebulize them, so they can use it as a delivery system. So you can use it as a delivery system. So instead of giving an IV injection, you can nebulize your whatever biomolecules it is, say for vaccine or DNA or anything, and you can just breathe it in. You don't have to go undergo a painful injection or an invasive technique. So it's a very cool system, and we have explored its potential into in various aspects like nebulization, and you can jet the fluid so that you don't have to use any pipette. You can avoid a pipetting error. Then you can centrifuge like concentrate particles that is dispersed in a liquid media, and concentrate them. So micro centrifugation kind of effects you can get. So these aspect, and even in material synthesis, so you can create a different types of materials using this acoustic stimulation. And later we found out that these acoustic stimulation can give rise to various aspects in biological systems, say for example adherent cells. So in adherent cells you can stimulate the cells and it give rise to a transient membrane aberration, through which can enhance the uptake of nanoparticles without any cellular damage. So unlike any other physical stimulation whose application give rise to around 40 to 60% of cell death, we can these stimulations are found to be more cell friendly, and you can get almost more than 95% of nanoparticle uptake without cell viability as high as 100%. So you don't kill the cell. So this is a very cool system. So while we were stimulating and studying this phenomenon, we found that it also affects the intracellular calcium level in the cell. And then we got an idea that maybe we can use this intracellular calcium to affect various number of aspects. So intracellular calcium is something can be considered as the center of all activities that is happening within the cells. So calcium can affect the cell viability, calcium can affect the apoptosis, calcium can affect differentiation, it can affect cell motility, how cells propagate, how cells move around, differentiate, how the cytoskeletal rearrangement happening. You can pattern the cells with the, even you can design its shape, how it should be spread. All those things you can um, uh, like manipulate, if you can manipulate the calcium stimuli, calcium intracellular calcium level. So difference in differentiation, these second messengers called calcium also plays a very key role. That's how we ended up with stem cell differentiation. So what we did was we got stem cells from bone marrow, um, and then we stimulated the cells with acoustic stimulation in two culture conditions. That is one basal media and another one osteogenic media. So in basal media, you don't have any chemical or biological stimulants, say like cytokines or dexamethasone, ascorbic acid, or anything like that. So basal media is just a media that is used for propagating the cells. It doesn't have any chemical or biological cues. And osteogenic media will contain chemical cues like dexamethasone, sodium pyruvate, beta glycerophosphate, sorry, not sodium pyruvate, ascorbic acid, and beta glycerophosphate. And we stimulated the cells for 10 minutes per day for 5 days. And we try to see whether we can direct the stem cell differentiation towards any of the lineages. And we found that, and we study different lineages like adipogenic lineage, chondrogenesis, and osteogenesis. And interestingly, we found out that when you stimulate the cells in basal media, it always direct the cells towards osteogenesis. So even without any chemical or biological cues, our acoustic stimulation can force the cells to choose only or go towards osteogenesis. So we so there are different osteogenic markers in the cells, and these osteogenic markers, how they present themselves at different stages determines at which stage the cells are. Say, for example, rungs 2, it is considered as the osteogenic determinant gene, where if you find rungs 2 expression quite high in the very early stage, you can um, kind of predict that these cells are going towards osteogenesis. And we found out that in basal media, without any chemical cues, with our acoustic stimulation, the cells were expressing rungs 2, and all these rungs 2s were, instead of in cytoplasm, it got translocated into nucleus, indicating that these cells are differentiating into osteogenesis. We have seen other molecules like call other protein of interest like col1a1, which is also it can also be considered as an early osteogenic markers. And so, in a normal, typical osteogenesis where you use only the chemical or biological stimulants, these rungs 2 and col1a1 expression was found around between day 7 and day 10. But with our acoustic stimulation in basal media, we can see the rungs 2 expression at as early as day 3, which means the cells are like a in a very fast mode when they are in mechanical stimulation in basal media. So you can get an early um, determine or early osteogenic lineage with acoustic stimulation in basal media. So when you have this regenerative therapy, we use different types of cells like iPSC, induced pluripotent stem cells, and normal stem cells. So when we use these kind of cells, it's always better to have an early differentiation, because there is a high chance that these committed cells can go back towards the not pluripotent stage or multipotent stage, or it can also hinder this or delay this complete differentiation. So it's always best to have an early induction of lineage, so which is what we can see with basal media. But in osteogenic media, what happens is you still get an improved differentiation, but it follows the typical profile, gene or protein profile, as that as something as you see in normal differentiation. So basically, you can understand that in basal media, cells responds to acoustic stimulation in one way, and in osteogenic media, cells respond to in different ways. So based on what cues you give, uh, so that's what the mechanical transduction is. So if you have a lot of other factors, then mechanotransduction is different. If you have just mechano cells that just have mechanotransduction to rely on, then it will behave in a different way. So, in order to understand that, what we did was we further studied what all protein profile the cells exhibit after complete 21 days. In 21 days, and we have seen that you can, even with just 5 days of stimulation, once the cells are committed, the cells finish that entire differentiation. You get a mature osteoblast by day 21, where which was marked with all the early and late osteogenic markers like osteopontin and osteocalcin, ALP. So basically, you just need a short stimulation instead of a long duration of stimulation, and you can bring about all these changes. So we wanted to understand how this mechanotransduction actually affects, why this is happening. So what we did was we studied key components like um, rock proteins, which affects the cytoskeletal rearrangement, and mechanosensitive ion channels like piezo 1 and 2 channels and TRPV channels. So, understanding how it works, understanding whether these are activated or not, will help us to understand how the our acoustic stimulation initiates the mechanotransduction. So from our studies, we found out that the cells, stimulated cells, have much rock proteins, and also it activates piezo channels, but it doesn't activate any TRPV channel. So basically, in TRPV channels are activated by, osmotic pressure, those kind of mechanical stimulation. But piezo channels are affected by any change in this membrane structure of the membrane deformation. So in the earlier studies, which I already mentioned, we have seen membrane aberrations with mechanical stimulation or acoustic stimulation. So piezo channels are activated. Rock proteins are basically that stimulate alters cytoskeletal rearrangement. So these two are molecules. These two pathways are activated with acoustic stimulation, indicating that it triggers MAPK kinases. MAPK induce signaling transduction, and which is why we find only osteogenesis and not any other lineages. But in uh, osteogenic media, we couldn't find any of these remarkable or significant change in any of these proteins, which means it kind of follows the paths that are osteogenic, normal osteogenic media without any stimulation follows. So we then wanted to see whether we can stimulate the cells, different stem cells from different sources. So bone marrow stem cells are pretty hard to collect. So we had stem cells from adipose tissue and umbilical cord, and we found out that even in those cells, stem cells from different sources, we can, although we just had to tweak the number of stimulation which we had to give. Just with 5 days of stimulation, we can still direct the cells collected from adipose tissue and umbilical cord towards osteogenesis. But we couldn't initiate any other lineages like chondrogenesis or adipogenesis from any of these cell source. So bone marrow cells are known to have a predisposition towards osteogenesis, and adipose cells, adipose-derived stem cells, are known to have a predisposition towards adipogenesis. And none of these cells, without any chemical cues, it couldn't go to its predisposed fate, that is like chondrogenesis or adipogenesis. It always stick to the osteogenesis. We are kind of yet to find, we are still pursuing why it is like that, and we are still trying to understand that. But this is so far we gained. And what we also did was, in order to upscale the process, what we did was we stimulated the cells for just 5 days and then reseeded them, like trypsinized and reseeded them into a different flask or plate, and we could still have this osteogenic potential of the cell. So once committed, the cells are not going back towards, or cells are not stopping their lineage right then and there. It can still go back to its osteogenesis or whatever lineage it has selected. So our system can be used, for a upscaling process. So you can just stimulate the cells outside the lab and then put it in a scaffold, implant it onto the patient. You can still get the osteogenesis. So compared to any other techniques, our technique can be considered as a supreme phenomenon. I hope that's it.
Well, thank you. I have a question a burning question to lead off with.
Yep.
Fascinating work, and I'm particularly interested in the mechanism of the mechanical coupling, and particularly in the megahertz frequency region. What interests me about that so much is, in a previous life, I did a fair amount of molecular dynamics of membrane proteins, and there are undulating modes, natural undulating modes that we would see in membranes, POPC, DHA, POPC mixtures, with membrane proteins embedded in them. So it's a curious thing. If you're pumping undulating modes in membranes, that would enhance the the temporal curvature and offer more opportunities for increased permeability. But it would also have an effect on, proteins, particularly ones that have evolved sensitivity to surface tension in the membrane. And I could easily see driving oscillations and modulating activity in those. Any thoughts on that?
Uh, we we didn't study much about membrane, effect of acoustics on plasma membrane as such. It's still like it's in its budding state. We are still studying that. We are getting a couple of collaboration to study the effect on the plasma membrane as such. But this acoustic stimulation, depending on the cell type which we use for the studies, it can have different effect. And the power which we use, the magnitude at which we stimulate the cells, can also affect. It have a specific effect on the plasma membrane. So if you're using a very low power input power, then it's just like a mere vibration of the cells. It doesn't give rise to any change in the cytoskeletal structure. But if you give an ample input, what happens is you create a transient membrane aberration. So basically, you can see a small hole that is formed when you're stimulating the cells, which kind of closes off, which closes off within, after cessation of the stimulation. So once you stop the stimulation, you can't see these kind of holes. And we are um, expecting, or our hypothesis is that when you create those kind of holes, it stretches the plasma membrane, and it affects the lipid draft, and then in turn it affects the, you know proteins like mechanosensitive piezo proteins or something similar like that. So I can say that it will affect the plasma membrane protein, but how far, how much, we are yet to study.
Oh, fast. So you're actually seeing pore formation in the membranes
Yep.
from. Oh, wow.
Yes.
Okay.
So there is a prior paper from our lab, Dr. Swati Rameshan's. So you can see, so what she did was she stimulated the cells and fixed the cells while stimulating, and then fixed the cells after 10 minutes after cessation of the stimulation, like 10 minutes after cessation of the stimulation. And she did electron microscopy, and you can see small holes at, uh, cells fixed at while stimulation. And you can even do a normal propedium iodide uptake studies with this, and you can see how it's affect the cells membrane, like a normal optimization thing, but you can clearly see that. So her paper will give that nice images.
Fascinating. Thank you.
Doctor, thank you very much for the talk, and this is absolutely fascinating. And I have a question. You mentioned that the nanomachinery, it's using the bioelectrical, body. It's using the human body as a power source to activate these little sound pulses. Is that correct?
No. AC current, so alternate
Oh,
current.
okay.
So we have a signal generator and an amplifier. So we give electric current to these IDTs, interdigited transducers, which is imprinted on. So if you have my paper, I think it's in figure 1.
Okay.
So in that, you have a piezoelectric chip, which is made of lithium niobate. So the and it has an IDT that is imprinted on this lithium niobate chip. And when you give power to this IDTs, it converts this AC current into mechanical energy. So this properties
Oh,
of piezoelectric chips are, it can convert electrical energy into mechanical energy, and mechanical energy into electrical energy. So that's the
ah,
property of this chip or
okay.
device
Thank
which
you.
we use. Yeah.
And one more question, please, is
Yeah.
how does this machinery target specific cells? Because I can't the size these things must be. How do they know? How do you direct them? Is it like do you observe them from the outside, or
Okay.
how do you?
So our system works only outside the body, like an in vitro condition where you have to take the cells out and plate it in a different so it's like an in vitro studies. You can't use it so far. You can't use it in inside the body. So if you have to target a particular cells, you have to get that particular cells onto a plate and then you stimulate the cells. And once you stimulate, you can put it back into what wherever you need. So we have couple of studies lined up for such like hardcore in vivo studies where you stimulate the cells and use the stimulated cells for further therapeutic effect.
Okay. Right, thank you
So
very much. Yeah,
yeah.
yeah, please.
So
Hey,
targeting basically you do with ultrasound, like low frequency where you can target. So you know the ultrasound scanning. So ours is megahertz frequency. Uh, so we can't target like that. Our range is bit low, around 500 micrometer. You can't go beyond that in the body. So anything on the surface we can manage. Rest of the thing you'll have to take the cells out.
Okay, okay, so that's probably still development down the line, possibly in the future. Thank you very much.
um, hey, Lizebona, thank you for this wonderful presentation. It's really interesting. So MSC cells can also turn into other things based on the signaling we expose them to. So why do you think that they are turning, or do you think you could use a different stimulation pattern to turn them into something else, or is this mechanical stimulation really um, calcium? Maybe it triggers the specific calcium signaling cascades that turn them only into into this osteogenic differentiation.
Yeah. So far, what we could pinpoint whatever phenomenon which we have seen to calcium and associated signaling cascades. We couldn't trigger anything else in, without any chemical cues. It's always towards osteoblast. So that signaling is very specific, which is what make our system very unique. So unlike any other system, our system is very particular that it triggers only calcium. So far, it is triggering only calcium related signaling. And calcium related signaling is majorly found in adipogenesis and osteogenesis. But um, this work is, there is another work which is not yet published, so I can't disclose a lot of it. There is another second messenger called CAMP. So calcium and CAMP kind of act together and it decides that it is only going towards osteogenesis. So we can't manipulate it beyond that. So far, we are not able to manipulate it beyond that.
Yeah, that's really interesting that the mechanic, because it's interesting from the basic, developmental, um, uh, insight. It's also interesting since for more translational aspects, women with osteoporosis, and so it's been speculated. I don't think there was like real mechanistic data to show that it's more beneficial to have impactful exercise to prevent osteoporosis versus like swimming and things like that. Do you
Yep.
think that that's the reason why?
So our stimulation imparts around 0 0.1 megapascal pressure on the cells. That is something that is that which is in the range what we experience in our body. So it could be the reason that it is kind of like some sort of predisposition will be there, and in the body that if the stem cells are differentiating into different lineage, it doesn't depend only on the mechanical stimulation, it has some biological stimulation as well. But when it is outside the body, you need to depend, our system depends only on mechanotransduction, and maybe that particular pressure activates only that particular signal cascades. So it's hard to predict, but I think it is something related to that.
Did you ever try pulling? Because we had a guest speaker here that talked about neural development, and she said that kind of tension mechanisms are important for neuron development. Serena, do you remember the um, about the zipping
I, I
during
don't
neuron
I don't
development?
remember that particular talk, but I'm very curious about environmental stimuli that I can't bridge the gap between what natural mechanical stimuli would be in the megahertz range, but it's interesting. And certainly, if there's tension coupling, that would certainly relate to medicine in space and space effects on skeletal development as well. But I don't remember that particular guest speaker.
It was the zipping. You found it very
Oh, the zipping.
fascinating. But
Yeah, yeah.
yes, there was also the tension that kind of pulling was also important for the differentiation.
Dr. Sargupta, I believe.
Yeah, exactly.
Yeah. Okay.
Yeah, it's really interesting that those mechanistic um, input or pulses inputs generate such a specific, like trigger a specific pathway for differentiation. I think that's really fascinating.
Yeah, we we were also mesmerized with this, but yeah, we are still continuing it. But no matter what type of cells we are using, it is always towards osteogenesis. We tried different other systems as well, but it's all osteogenesis. Without any chemical cues, it's all osteogenesis.
So, are you done,
Yeah, yeah,
Katerina?
go.
So, thank you so much for sharing your fascinating work. I mean, I really enjoyed that. So, to me, message out of that was applying the right amount of the frequency or pressure or whatever in the right places to the stem cells, how it can trigger the change processes. But what is came to my mind as a question for the future, I mean, application was the implication of the bone marrow adipose tissue on inflammation somehow, how it can be related with the research that you did so far. Because as you said, that it's very hard for, predicting how it can work. But I think that it can be a good research proposal. I don't know.
Bone marrow related inflammation, is that what your question is?
Yes, bone marrow adipose tissue.
Okay.
So we know that it can be related with the inflammation, and we just consider that in too many different, lineage from the bone marrow, also in myeloid process, and for example in the multiple myeloma, they have attention on it, and I was just wondering how we can apply your research on that. So that was in my mind. I just wanted to share with you and see
Yeah,
your opinion over.
I'm not I'm not sure how we can apply this, but um, anything that is related to calcium signaling, um, if we can do. So we have a couple of other works that is lined up, and we are doing it. So if any of the cellular process which we you are interested in can have some relation with the calcium signaling, uh, um, I'm hoping that we can bring about that change in the system. If it is related to calcium, acoustic stimulation can bring that. I'm not sure how I can put my research into the scenario which you have asked. Sorry.
I actually have one more question for you, Doctor, if I can. In the abstract of your paper, I saw it mentioning that there's been an assertion that there was little point in applying sound waves above is it 1,000 kilohertz? Um, why was that assertion made before? You found something to the contrary, but why did they think that it was pointless to go that high?
So they when they did stimulate the cells to more than 100 kilohertz, they couldn't see any difference in the osteogenic potential of the stimulation of the cells. So they said it is it just requires something that is beyond 100 kilohertz. So that's what they suggested, but we showed them that it's not just it's not just the kilohertz or the frequencies that we uh, need to care about. Ultimately, it's how much the cells are receiving. So um, with our stimulation, the cells receive exactly what our body is experiencing, the cells in our body is experiencing, which is why we are able to bring about these changes. And those low frequency stimulation, they you need to stimulate the cells for a very long duration, like they stimulate the cells for almost 21 days, uh, which is not and which is how they are imparting that or triggering that signaling transduction. So you don't need to stimulate that much longer if you can trigger the cells at the right amount. So our megahertz frequency can trigger within a short period at the right amount so that that signaling cascade can start. Theirs requires a longer duration. So they just studied with 1 kilohertz, nothing beyond 100 kilohertz. I mean, with whatever they recommend, they couldn't bring about those changes which we see.
That's fascinating, and it's really exciting to see, when an assertion like that can be challenged and more progress can be made. Thank you very much.
Yeah.
One additional question. So, causal signal can be a lot of different things. So, you discuss in the paper that later on that the MAP kinase ARC1 differentiation activates the RUNX2. So that's the main pathway you think that is being activated through the stimulation?
Yeah, so in basal media, it's MAP kinase pathway. In osteogenic media, it's Wnt pathway. So depending on the number of cues or types of cues the cells are receiving, cells opt different pathway. That's the key thing to take in as a if you are interested in mechanotransduction studies. So it's not just one stimulation. Depending on number of stimulation cells received, cell can switch on n number of pathways.
But then it leads to the same cell type. That is so fast. Like, it puzzles me. That is so interesting. And then you have all the different stem cell types that still lead. Do you see any differences? Like, depending on which pathway you elicit, do they later on have any type of, I don't know if you later on that maybe, or you're doing like RNA sequencing or I don't know, more histology. Like, is there a difference later on? Like, are they maybe, I don't know, from morphology or gene expression
Oh,
profile different?
okay. So the key thing is depending on the tissue source or the cell source, we have to tweak the treatment a bit. Like stem cells from bone marrow were pretty susceptible to our stimulation. And while umbilical cord derived stem cells and adipose derived stem cells, we needed to trigger them a lot more in comparison. We and multiple number of times in the same day to evoke these signaling cascades. So I think depending on the sources, the cells receptors that receives the signals changes, the cytoskeletal arrangements changes, which is why we needed to tweak our stimulation a bit differently to bring about these changes. Morphologically, they all look the same. If you are looking at the protein profile, bone marrow derived stem cells had the best osteogenic marker expression compared to other two. That's probably because these cells, adipose derived and UCSCs, are bit more predisposed for other lineages. So we are forcing the cells, which is why that expression level is a bit less, but it's still enough to trigger osteogenesis.
And on top, you write in the end that the fabrication cost per device is really cheap. It's
Less.
$1.
Yeah.
That's so amazing. That's
So you
really
can
cool.
upscale it pretty quickly and pretty easily. It's not a huge device. You don't need a much expertise in the system. A 1 week you don't even need a 1 week training. A 1 or 2 days training is enough to run this system at all. So it's very easy, very simple system.
Yeah, the only problem with upscaling is that you cannot transplant it from one person to the other. Are you working on that too?
No, so in this paper, what we did was we stimulated the cells for 5 days and then trypsinized and reseeded them onto another culture plate, and we could still get osteoblast at the end of 21 days. So which means that whatever cells are committed with our stimulation, it still remain committed even after reseeding. So that signal transduction is still there. You don't So basically you can uh, use the system as a pre-treatment. Say for example, in clinical studies, what we do is we have a scaffold onto which we have our stem cells. We seed our stem cells and then trigger the cells, like chemical cues are given or biological cues are given. And then once it is almost differentiated, you put that in the patient. So that's what we do in the clinical trial so far. So with our acoustic stimulation, you take out the cells of the patient, trigger them outside, like in the lab. And after 5 days of stimulation, on the 6th day, you trypsinize them, reseed them onto the scaffold which you want, and then you can keep it place it in the patient itself, and it can theoretically, as per our studies, it can still have that osteogenic potential. So you don't need to have a 21 days of stimulation even with chemical stimulants, and you can still get it. So we are planning to do something similar to that. We have a collaboration with Sense Lab ACMD, and we are planning to do it. But yep, it is possible. You can upscale it and you can translate into the more clinical scenario.
That's amazing. This will cut down costs quite significantly and make it more
Yes, we
affordable.
can.
That's
Yeah,
amazing. Congratulations on this wonderful paper. And
thank
yeah, if
you.
anyone has more questions, please go ahead. The hour is almost over. So if you have any more questions, please come to the stage. Yeah, I think this is really cool research. So are you working also on the more clinical study, or what are you doing next? What's in your future?
Yeah, like we are still planning to put this work into so that we can achieve something around translate into clinical studies. That is one thing, and I'm also I'm more interested in mechanotransduction. So I'm still doing couple of things with calcium and other second messenger involvement with acoustic stimulation. So that is also there.
Yeah, wonderful. Yeah, if nobody has questions, Victoria?
Yeah, hi. This is really a general question in a completely different direction, but I'm just thinking of applications of your work in terms of I'm always concerned about the effects of sonic testing in the ocean on ocean life, cetaceans, and I'm thinking, I was always concerned about ultrasound, during pregnancy, and so I'm thinking about this in terms of the Navy, U.S. Navy doing sonic testing and what that means for for anybody who's living in the ocean, and
Um,
yeah.
yeah, unfortunately, our megahertz frequency, it doesn't travel like that. Uh, Waves can't travel more than a particular. It is restricted to that surface. So, unlike ultrasound, which can travel a long distance, our system can't travel more than 500 microns. So, yeah, application is very restricted. So I don't think we can use. I'm not sure whether we can use in this navy or something aquatic related, unless and until you can think of something that can be done inside the lab and then translate.
Yeah,
Um,
thank you.
I have I have one last question for you, and please forgive me if I've misunderstood a part of this, but um, when you mention that sonic waves are, you can use them to, make it a differentiation in a certain way. Are you hoping that in the future you discover how to also use these sonic waves to choose multiple options of changes rather than just the ones you're describing at the moment?
Um, so far, we didn't have any success with other lineages, so we need to figure out exactly why it is happening and then come around another treatment modes. So yeah, in future, but not in near future.
But it is something that you would have your eye on later on.
Yeah.
Fascinating. Thank you.
So we
Thank you
have
very much.
tried like initial trial, it failed. So it acoustics always prefer osteogenesis. It always prefers bond cells. So we need to figure out
Hmm.
why exactly it is and then
That
come
would be
around with
really
something.
interesting to find. I think if we found the answer to that, it would
Oh,
explain a whole lot more. Thank you very much.
yeah, that's fine. Yeah. Thank you
Also,
so
there are
much
a
for all the
couple of
questions.
questions. Yeah, there are a
Yeah.
couple of questions in the chat. I mean, if you can just have an access and you might have some responses to them, that would be wonderful.
Okay. Calcium signaling in SARS-CoV-2.
I
Is
think
that
the questions
sympathetic?
don't really, they don't really have anything to do with the research here,
I
because
can't.
the sympathetic system is not involved here. This is cells in media that you take out and stimulate specifically while you grow them in media.
Yeah.
It's not in the body. Yeah.
Okay.
And then the COVID, I don't. think it has anything to. A lot of things use calcium signaling. Neurons use for different things. Calcium, like a lot of things in the body use calcium
Yeah.
signaling. It's just that then what the signaling cascade, which one gets triggered by a very specific calcium signaling transmitted by very specific receptors, that is then what triggers like these specific pathways, and then differentiation in this case into these cell types. Sorry, I was just answering for you because I was reading about it and like I was reading the questions
Yeah,
in
thank
the
you. Thank you.
You're welcome. Okay, thank you so much for
Yeah,
your
if you
time.
have any questions, just shoot the questions to the email. Either Leslie or Amy, they'll always direct the, or even in this clubhouse, I can answer it. I'm more than happy to answer your questions if you have any questions after going back home.
Okay. Yeah.
Thank you
Yeah.
so
Dr.
much
Gig
for that.
says he means sympathetic resonance of the sound.
This is
I'm
sympathetic
not.
resonance, bone minerals. Yeah, I'm not sure what.
Yeah.
It
I
would be great if you can.
maybe Dr. Gig, look in the method section the specific wavelengths we were talking about, and the specific device, what type of sound, basically, or mechanistic pulses it's generating. Maybe that answers your question. Or what do you think, Lizebona?
I didn't get the question. Uh, rather than, yeah, I didn't get the question properly. So I'm sorry, but if you can explain it a bit more, I will be happy to address it.
Okay. Yeah, perfect. Yeah, again, thank you so much. If you can share updates with us soon again, please come. Your work is so interesting and also very important for the future of regenerative medicine. So yeah, thank you for working on this. It's a wonderful paper. Congratulations.
It's incredible,
Thank you
Doctor,
so
and
much
working
for
with
the feedback.
sonics is incredible. So interesting. Thank you so much for coming.
Yeah, really
Thank you so much.
fascinating. Thank you so much.
Yeah, thank you. Thank you.
Yeah,
Yeah, thank you, Lizebona. Thank you, friends in the audience, for coming and for keeping the room chat so rich with your questions and comments. We really appreciate all of you for being here.
yeah, and if you like this discussion, follow Science Society. We will have more guest invited speakers this week. Tomorrow we will have a group from South Korea, Dr. Jim et al., talking about quantum charging, to quantum charging of batteries. And then yeah, we will have about how the human vision reconstructs time. And then we'll have a room on Friday about building blocks of life and how they form, how peptides form on space dust, from Max Planck Institute in Germany. So yeah, thank you so much, Lizebona. Special thanks to
Yeah.
you. Thank you, everyone, and enjoy the rest of your day, evening, or wherever you are. Bye, everyone.
Thank you,
Bye.
everybody.
Thanks,
Good
everyone.
night.
Bye.